Okay, we finished talking about digestion and absorption of protein. Now we go to digestion and absorption of fat. So B, protein. C, fat. We first talk about bile salt. So where does which organ make or produce bile salt? Well, of course we repeat it many times. It is our liver. And what is the molecule that the bile salt is made from? If you remember our endocrine, is it endocrine? No. If you remember the early, the first exam, before the first exam, the first module of the lecture, we talk about the 4 major biological molecules. When we talk about fat, when we talk about lipid, we talk about fat and oil, first one, and then phospholipid, the second type. And then we talk about the third type is the cholesterol, steroid, the third type. Steroid is the third type of the lipid molecule. Phospholipid, steroid, and then one more. I don't remember. We have 4. We talk about 4 different types of fat and lipid. 4 different types of lipid. First one is fat and oil. Second one is the phospholipid. Third one is steroid. I forgot the last one. Sorry. But the bile salt is made by cholesterol. Cholesterol is under the category of steroid. It is under steroid. And after we make the bile, bile is stored in gallbladder. So what is the function of bile? Function of bile is to break down oil droplet, a big oil droplet into small oil droplet. So I always compare bile to detergent, dishwashing detergent. They are basically the same. We call both this dishwashing detergent and bile, we call them amphipathic. Amphi is like amphibian. Amphibian is the animal that live in 2 environments. So amphi meaning 2, pathic meaning that 2 affinity, 2 pathway. What are the 2 pathway? Hydrophilic and hydrophobic. So this molecule is both hydrophilic and hydrophobic. Bile is both hydrophilic and hydrophobic. Let's take a look. So this is a biomolecule. As you can see that it has a basic structure of a steroid because it is made from cholesterol. And this is the, you can see that it indicates the hydroxy group. Hydroxy group, they are all in on one side of the molecule. We call it polar side because hydroxy group, the OH, makes the molecule love the water. It can interact with water, and on the other side is the non-polar side. This side can interact with oil and fat. So one is like someone that on the non-polar side is holding hand with the oil and fat. On the other, but the other hand, it is holding hand with water, so that it can do this. So you have a fat globule. The process is called emulsification, meaning that we add bile salt so that the non-polar side of the bile will surround the fat so that it will break down the fat into small droplets, like this. The polar end will be facing out to the water. So the process of breaking down a fat globule into small droplets, we call it emulsification. It is exactly the same mechanism of your dishwashing detergent use. But this washing detergent is also amphipathic, so that one end you will hold the oil on your plate, the other end you will hold the running water from your tap, so that when you rinse, after you apply detergent, when you rinse your plate in running water, then the detergent can pull the oil away from your plate. What is the purpose of this? We are not washing dishes, okay? What is the purpose of emulsification in your digestive system? Think about this. When we break down a gigantic fat globule into a small droplet, what do we increase? What does it do? We actually increase the surface area of the droplet. When we increase the surface area, what does it enhance, or what does it improve? It improves the it provides a larger surface area for the enzyme lipase to break down the fat, break down the oil droplet into glycerol and fatty acid. So that's the main purpose. The main purpose of emulsification is to increase the surface area of the fat and oil, so that lipase has a larger surface area to break down fat and oil into glycerol and fatty acid. And in other words, emulsification is very similar to mechanical digestion. Remember mechanical digestion, we have an example in the mouth. Chewing, using our teeth to cut the food into small pieces, is a form of mechanical digestion, because when you cut the food into small pieces, it increases the surface area of the food, so that the digestive enzyme have a larger surface area to break down the polymer into monomers. Similarly, emulsification is very similar to the mechanical digestion. The main purpose is to increase the surface area of the oil and fat, so that enzyme lipase will have a large surface area to break down the oil and fat into glycerol and fatty acid. Okay, now let's continue with our lecture notes. Amphipathic. It is for emulsification. Emulsification is for increasing the surface area. For digestion. So here we have the fat. Let me see if I can finish the pathway here. Fat. Will be digested by lipase through hydrolysis. And then you have a fatty acid and glycerol. And then bile salt. No. Okay.